Good afternoon. Welcome to the Wednesday NIH Directors Lecture Series, and today is the Margaret Pittman Lecture. I'm Mary Kay Floater from NINDS, and I have the great pleasure today to introduce to you two remarkable women scientists. And before I introduce our speaker today, Dr. Susan Lindquist, I'd like to say just a few words about Dr. Margaret Pittman, for whom this lecture is named. Dr. Pittman was the first woman to be a lab chief at NIH. She was chief of the Laboratory of Bacterial Products and became that lab chief in 1958. She had come from a small town in Arkansas, just a little bit west of the Ozarks, and her father was a country doctor, and she attended rounds with him on his patients. She went on to the University of Chicago, where she earned a degree in the very new field of bacteriology. And she also then went on to the Rockefeller University, where she did some very seminal work showing that the pathogenicity of Haemophilus influenza was related to the polysaccharide moieties on their surface coat. And she identified the 6 serotypes of H. flu, labeling them A through F. And I think we all know H. flu B as the primary bacteria that causes childhood meningitis. After that, she came to the NIH, and apparently she was a little bit unemployed during the Great Depression, but there was this sort of stimulus package from Social Security, and so she was able to come here. And she did quite a bit of work that was important for the production and standardization of vaccines for pertussis, cholera, and typhoid. She was here actually for 52 years, and the year before she died, they established this lecture series in her honor. And the lecture is given each year by a researcher who is a leader in their field, who demonstrates success through the example of her own science, and who is dedicated to improving the careers of women scientists. So since 1994, when this lecture series began, every speaker has exemplified the intelligence, drive, and scientific excellence that made Margaret Pittman a leader at NIH. So our speaker today, Dr. Susan Lindquist, certainly exemplifies those qualities. She's a superb scientist and a leader, and her contributions have influenced many fields, and in human disease, she's made contributions in neurodegeneration, cancer, and infectious diseases. She's a marvelous communicator and a great mentor to her students. Dr. Lindquist earned her Ph.D. in biology at Harvard and then went to do postdoctoral work at the University of Chicago. Her work on the heat shock response in yeast and Drosophila was highly influential and very cited. I think she has one of the most cited papers. And she advanced up the academic ranks there at the University of Chicago to full professor. But she was lured to Boston and became the director of the Whitehead Institute, as well as a professor of biology at MIT. Continuing her work on heat shock proteins and the degradation and misfolding of proteins led her into the fields of neurodegeneration and carcinogenesis that she's going to talk about today. She's currently a member of the Whitehead Institute, an associate member of the Broad Institute, the David Koch Institute for Integrative Cancer Research, and on top of all of that, she's also a Howard Hughes investigator. Dr. Lindquist.
Thank you very much. It's great to be here. I've had a wonderful day. I want to thank everyone who was involved with this visit today. I really appreciate it. And you might notice that the title of my talk has changed a little bit. It's evolved a little bit. The reason is that when I started trying to find out a little bit about Margaret Pittman and started reading about her career and the fact that she was involved in infectious diseases, I thought, well, we're now in my lab working on some infectious diseases, and I thought, well, maybe I'll tell you a little bit about some of that too. And then it turns out that it's the 200th anniversary of Darwin's birth and the 150th anniversary of the origin of the species, and we're working on evolution too. So I thought I'd step back a little bit, and I thought, well, maybe what I can do is give you a view of the heat shock response and protein folding in cellular systems that kind of is a broader view than I usually give. And I hope that this is going to work. I hope it won't be a kitchen sink, everything but the kitchen sink kind of talk. And I'd be most happy to hear from you afterwards if you think it was too much. So I work on this general problem of protein folding. And why is protein folding a problem? Proteins start out as long linear strings of amino acids. And in order to function, they have to fold up into these very complicated, intricate, very, very specific shapes or they don't work. And sometimes if they misfold, they can actually cause toxic effects. And the problem with their attaining those folds is that they have to do it in a really ridiculous environment. They have to do it inside of a living cell. And the reason why that's a ridiculous environment is that the concentration of other proteins inside of a cell is 300 milligrams per mil. That's just a staggering concentration of proteins. And so as a protein is still being translated, it's being bombarded by other proteins all the time because proteins inside of a living cell have a tremendous amount of energy. There's a lot of kinetic energy. They're actually really moving around very fast. And so when you think about proteins going about their business inside of a living cell, it's really not like Fred Astaire and Ginger Rogers. It's much more like the characters in a Marx Brothers movie, where chaos is poised on the precipice of disaster. So we call that in the protein folding field going off pathway, and it happens all the time. So constantly in living cells, proteins are misfolding. And the cell has a wide variety of mechanisms to help cope with this problem and actually uses quite a bit of its cellular energy to do so. And these solutions to this problem are extraordinarily conserved across virtually all organisms. In fact, I would say most of them are conserved across all organisms. And so we often wind up working actually moving from one organism to another because it has a particular advantage for studying some particular problem. And we work on molecular chaperones, protein remodeling factors, osmolytes, degradation machinery, and cellular compartmentalization. And all of these are mechanisms by which the cell helps to cope with this problem of protein folding stress, which, as I mentioned, is going on all the time. And then when the organism is stressed, and most typically by a heat shock, which is what we employ in the laboratory, but many other kinds of stresses, low energy metabolism even causes protein folding stress because all these mechanisms require a lot of energy. Changes in oxygen concentrations, changes in heavy metal ions, many things will induce additional protein folding stress. And what I'm going to be telling you about today is that this is so intimately integrated with the biology of an organism and the biology of the individual cells that it's really, we think, playing a huge role. And as I mentioned, we're going to start out telling you a little bit about some infectious disease issues. Also, we think it might be playing a very large role on the balance between neurodegeneration and cancer. There's been quite a bit of epidemiological evidence that people who get neurodegenerative diseases don't get as many cancers as you'd expect and vice versa. And we think HSF is one of the factors, HSF being the heat shock regulatory factor that regulates the stress response to protein folding stress, plays a major role on that axis. So what's the heat shock response? Well, here's an example. We took 2 aliquots of a yeast culture growing at 25 degrees, and we shifted them both up to 50 degrees for a few minutes. The difference between the two was that this aliquot here first got a half-hour pretreatment at 37 degrees. So just half an hour at 37 degrees provided an incredible increase in the ability of this cell to survive high temperatures. And this response is conserved across all organisms. Very, very ancient response. This is another experiment we did with the Arabidopsis seedlings. Basically, they were both treated at, the seed beds were both treated at high temperatures, but in one case, and this is pretty obviously this one, they were first given a couple of hours of a mild heat pretreatment, and that induced an ability to cope with this extraordinary stress. And while the cells are undergoing this protective response, they're making a number of proteins called the heat shock proteins. So these are cells labeled at 25 degrees, and these are cells labeled at 39 degrees. And you can see, which is a heat shock temperature for a yeast cell. And this response, as I mentioned, is induced by many, many other stresses, and it's very, very highly conserved. Moreover, the regulation of this response is even very, very highly conserved. The master regulator is called HSF, heat shock factor. And Carl Wu, right here at NIH, has done a lot of work on that. And that's the same factor that regulates this response from yeast to man, and in fact, most of these proteins are conserved from yeast to man. Not all of them, but most of them. And each of these proteins, it turns out, winds up doing a rather different thing to help cope with this protein folding process. For example, HSP104 is involved in actually taking aggregates of proteins that have aggregated under the stress conditions and takes them apart. HSP90 is involved in chaperoning a wide variety of particularly unstable proteins. I'm going to be telling you specifically, starting out telling you about specific functions of HSP90. And then HSP70 is a chaperone that binds to unfolded proteins, little hydrophobic pieces that should normally be sequestered inside the protein, and it prevents those things from getting aggregated to other proteins. So just as an example, it's a very multifaceted response, lots of different strategies employed to help cope with protein folding homeostasis. The first thing I'd like to tell you a little bit about is that some work that we've been doing on HSP90 and the role that it can play in the evolution of new phenotypes. We actually first started working on this in Drosophila and then moved to Arabidopsis, but I'm not going to tell you about that work. I'm going to tell you about some early work we did in yeast and some more recent work we're doing in both fungi and in cancer that has to do with progression, the evolution of new phenotypes in terms of oncogenic transformation and the evolution of drug resistance. So let me give you a little background on HSP90. As I showed you, it's induced by heat, and it's induced by heat in yeast and every other organism. It's essential for life, important protein. Very abundant even at normal temperatures. You normally don't need very much, however. You do, however, need much more protein under conditions of stress. So the cells can live perfectly happily with about 10% of their normal HSP90 levels at normal temperatures, but if you try to grow them at higher temperatures, they'll die unless they have that full, massive, additional amount of HSP90. And this fact that you make much more of it at normal temperatures than you need actually turns out to really, really matter in the two things I'm going to mention to you, the evolution of drug resistance and the evolution of evolved malignancy, and also matters in terms of therapeutics, because some of you may know that HSP90 inhibitors are now being widely tried in clinical settings for a wide variety of different malignancies. And it's because there's a buffer of excess HSP90 that the cancer cells need to use, but our normal cells don't need to use that excess 90, so you can inhibit it and have a therapeutic index. So this work goes back a long, long time when we were first working with yeast cells and first figuring out how to reduce HSP, how to regulate heat shock proteins and how to set up genetic strains at different levels of heat shock proteins to test their functions. And we had those genetic results that it was essential, you needed more for stress. It didn't tell us too much about the function of the protein. And then we were very lucky that just as we were doing this, two other groups of laboratories, actually several laboratories all within the same year, wound up running into HSP90 by complete accident. So one group was that was involved with looking at steroid hormone receptors, and they found that HSP90 was complex with the inactive form of the receptor. And another group was looking at oncogenic kinases which have been mutated cause cancer, and they found again that HSP90 was complex with the inactive form of the kinase. So it was postulated that HSP90 was a protein repressor, like there were DNA repressors that bound to DNA and repressed it. The idea was that this was going to be a protein repressor. So bind to HSP90 and HSP90 was going to bind to these proteins and repress them. And so we had just set up these cells in yeast where you could lower the levels of HSP90, and there was a very simple prediction. If HSP90 is a repressor, if HSP90 is a repressor, then when we reduce the levels of HSP90 in the cell, these proteins should become hyperactive. And I'm not going to, we actually first started out working on this with steroid receptors with Keith Yamamoto's group. I'm going to skip that. The story is basically the same as with VSARC, but VSARC relates to this cancer thing I want to talk to you about. So I'll just skip right to VSARC. So how did we test that? Well, we simply put VSARC under the control of an inducible promoter, and we put it into yeast cells. And we found that when we looked for SARC accumulation, the SARC protein accumulated at the same level in the wild-type cells or the cells that had these reduced levels of HSP90. There's a little difference, okay, but not a big difference. But when we looked at the activity state, it knocked our socks off. I mean, it was a huge difference in activity. However, it went the exact opposite direction. When you reduced HSP90 levels, you had no kinase activity whatsoever. So we first got into thoughts about evolution in HSP90 with this experiment because we decided we would compare the oncogenic kinase with the unmutated normal cellular form of the kinase. And when we put that in, well, we had to expose that for much, much longer because it's a much less active kinase. The mutation that activates VSARC makes it hyperactive. But as you can see, when we did expose it for longer, there really was only a small difference in the activity state of the cellular kinase when we lowered the HSP90 levels. So what happens is that CSARC is normally a self-inhibited kinase. It folds back upon itself and inhibits its own activity and only signals when it's supposed to signal. The mutation eliminates that self-auto-repression. It makes the protein hyperactive, and that's what causes us to have oncogenic properties and signaling in a deranged manner. But that same mutation, which confers this new property on the cells, also causes the protein to misfold. It doesn't fold very well. And so it turns out that what happens is that HSP90 acts as a chaperone. It recognizes this protein as a protein that's having trouble folding, and it helps the protein out, much to the detriment of the organism, but to the benefit of the cancer cell. And it helps promote its activation. Now, while HSP90 is bound to it, it's keeping the protein inactive, but the purpose of HSP90 is not to act as a repressor. It's to help get this protein into an activatable state in the first place. And that's basically the same way it works with steroid receptors as well. So it promotes activation. Now, I went to the University of Chicago Intellectual Property Office at this time, and I said, well, this is going to be really great. It's going to be a great target for cancer therapy. And they were rather rude, so I could say it. They thought I was crazy. We'd taken an oncogenic protein and brought it into a yeast cell. And at about that time, however, Luke Whitesell had heard about our work and was doing some other independent work on this interesting molecule called geldanomycin. And he found, in fact, that this same principle holds true in mammalian cells. And this was work he did with Len Neckers here at NIH. VSARC transformed cells grow like crazy. They're not contact inhibited. If you use this compound called geldanomycin, which turns out to be an HSP90 inhibitor, although it wasn't known at the time Luke was doing this work, it reduces the levels of functional HSP90 in the cell, it prevents the activation of VSARC, and the cells return to normal morphology. And is this relevant to humans? As I mentioned, it sure is, because these are just a partial list of the companies and the trials that are going on with HSP90 inhibitors in a wide variety of oncogenic conditions. Again, taking advantage of the fact that normal cells don't need very much of this. It's in the oncogenic state that you need so much protein. So the first way in which we found that HSP90 plays a role in the evolution of new phenotypes is that it chaperones mutant proteins. I'm going to tell you about one more mechanism. There's many others. It's really amazing how multifaceted the role of this protein is in terms of the evolution of new phenotypes. And if you're interested in evolution, it's a story worth following. So what we now know about HSP90 is that it binds to a wide variety of really quite different proteins. As I've already mentioned, steroid hormone receptors and kinases, which really don't have anything much in common. What they have in common is that they have one domain that's generally well-folded and another domain that's very unstable. And it's meant not to finish, these proteins, its clients are generally meant not to finish folding until something else happens. They bind a partner protein, or they bind a ligand, or they get phosphorylated and inserted into a membrane. And these proteins, it's a minority of proteins in the cell, but they're really important proteins. They're proteins that are involved in the regulation of all sorts of important processes. So if you look at oh, by the way, this involves many, many co-chaperones. And it also involves, there are many variations on this general thing. This is kind of hand-waving, but it's in loose outline, it's correct. So if you look at a signal transduction diagram of a cell, and I just borrowed this one from Hanahan and Weinberg, and ask where on this does HSP90 function? As you can see, it functions all over the place. And it's this functioning in the circuit diagram that actually influences the next role of HSP90 in evolutionary processes. It's protein folding and evolution. And basically what I'm going to tell you about is that mutations can create new phenotypes in another way, not by specifically giving a protein new function, but by altering the way a cell is wired. And that the cell needs to respond. Those mutations can give a beneficial phenotype, as you'll see in a minute. But they also create their own problems in the cell in terms of protein folding and the normal protein homeostasis. And that means that those cells will go from making much more HSP90 than they need to barely having as enough HSP90 as they need, because they need much, much more protein to cope with deregulated circuitry and help the cell to reach a new homeostatic balance. So this involves drug resistance in fungi, Saccharomyces, Candida albicans, and Aspergillus. Fungi separated by 1 billion years of evolution. I'm going to step right in. We actually did most of our work in Saccharomyces cerevisiae, but I'm going to step right into something more medically relevant, and that's Candida albicans infections. The evolution of fungal drug resistance is a huge problem. It's ubiquitous. It happens all the time. And we wondered whether HSP90 might be playing a role in this for a wide variety of other reasons, other experiments we've been doing, and actually in Drosophila and Arabidopsis. And in the laboratory, at least it did, when we had Candida albicans incubated in the presence of fluconazole, you can see that they very easily evolve drug resistance. But if you add a level of an HSP90 inhibitor that just takes away that excess buffer, doesn't inhibit it completely, but just takes away the excess buffer, cells in the absence of the drug are just fine. But in the presence of the drug emergence, the drug resistance does not emerge. It's a profound difference. Now, there are some mechanisms by which cells can evolve drug resistance that don't involve HSP90, but an awful lot of them do. So that gave us the opportunity to actually look at evolution in the human host, not by infecting anybody with the fungus just to see what would happen, but rather by taking advantage of the fact that Spencer Redding and Ted White and colleagues had been working with patients that were suffering from fungal infections and took serial isolates from those patients. And genetic analysis of them indicated that it was very interesting. It wasn't when that person was treated with an antifungal and wound up responding and being okay for a while, and then coming down with infection again. It wasn't a new infection. It was a reemergence of the pre-existing pathogen. And so they have made a whole series of isolates from several different people. We've tested several of them, and they all work the same way. So basically, what we have here is just green means growth, black means not growing. And these are fluconazole concentrations. And then along here we have first our lab strain, which is very sensitive to fluconazole, and then subsequent isolates from this host over various periods of infection over the course of 2 years when the unfortunate individual succumbed to this particular fungal infection. These fungal infections are deadly. As you can see, even the very first isolate is quite resistant. The later isolates get even more resistant. So now we were able to ask not what happens in a laboratory, but what has already happened in this human host. Has this fungus used its HSP90 buffer to allow it to be resistant to the drug? So we just used that HSP90 inhibitor. Actually, we used 2 structurally distinct inhibitors. And we've now done it genetically, but it really was hard to do genetically in Candida. And as you can see, a small amount of HSP90 inhibition, taking away that excess buffer, completely abrogates the resistance that this fungus has evolved in this natural war with its being host and pathogen. Now, as I mentioned, you could partly see it here, but it's very clear here, that eventually the fungus has evolved to the point where it's resistant even to the HSP90 inhibitors. And so one might wonder, well, what the heck was happening there? Because after all, this person was not being treated with HSP90 inhibitors. Why did resistance evolve? And we think it's the tie-in with protein folding homeostasis and natural environmental stresses, in this case, for this fungus, likely we think fever. Because we find that when we simply grow the fungus at 39 degrees, that has greatly already compromised the ability of it to survive in the presence of this drug. And that's, again, because under conditions of stress at higher temperatures, the organism really needs that full buffer, that full absolute amount of HSP90. So we know a lot about this mechanism of resistance, and I'm not going to go into any detail. The drug targets an essential sterol of the membrane, and they block this pathway, wind up producing a toxic sterol that would kill the organism, but they can acquire mutations, for example, further upstream in the pathway. This is a little oversimplification, but it's basically the idea that cause a different sterol to accumulate in the membrane. And now this organism can limp along. It's actually can grow pretty well. But the only reason it can grow pretty well is because it's got its circuitry involving calcineurin and membrane stress turned on like crazy. It's now using that buffer of HSP90 to mature this massive pool of calcineurin, which it normally does not need to mature in order to survive this stress. So this is a very different way that HSP90 is involved in the evolution of a new state. It chaperones responders to stress, responders to the effects of mutations that simultaneously solve but also create their own problems. And this, we think, is very broadly true. I mentioned that we tried it in Saccharomyces cerevisiae. We've also looked in Aspergillus, which is about 1 billion years separated from Candida. It's a major human pathogen. Mortality rates approaching 90%. Once you're infected with Aspergillus, you're in bad shape. And resistance to an entirely new class of antifungal, echinocandins, has already been emerging in the clinic. And when we asked whether those strains of Aspergillus that have acquired antifungal resistance, drug resistance, are using their HSP90 to drive that resistance mechanism, they are. So these are strips containing different concentrations of the drug, and you can see they're completely resistant to these are echinocandins. They're completely resistant to the echinocandins. These are 3 different clinical isolates. But if we have them growing in the presence of a small amount of HSP90 inhibitor, it completely abrogates that resistance which they have naturally evolved in the clinic. So, okay, so that's just giving you some details about just the ways in which one particular heat shock protein interfaces with human health and biology in terms of infectious disease. So what I'd like to do now is talk to you a little bit more about the heat shock response as a whole and how that might interface with human health and disease. So I'm going to talk to you about the master regulator of the response, heat shock factor, which, as I mentioned, is conserved from yeast to man. And now we're really talking about this entire response. In mammals, this heat shock factor regulates only the induction of the heat shock proteins in response to stress. The normal cellular levels that are present under normal conditions are not affected by HSF. Those are other transcription factors that regulate that. And what I'm going to be telling you about, we think involves these massively synthesized major heat shock proteins. But I just want to mention that in the early days when we were working on this heat shock response, we looked very intensely at these proteins that are made in massive quantities. And it turns out, as I mentioned, they're made in massive quantities to cope with this massive problem of protein misfolding that's already a problem, and it gets worse with stress. But I want to mention that in addition to these things which kind of knock your eyes out with the way they're made, there are a large number of other proteins that are synthesized at lower levels, all kinds of very interesting molecules, kinases and apoptotic factors and all sorts of interesting things that are part of this response. You just don't really notice them very much on the gel. And we think, so I'm talking about this whole gestalt of a response. And it turns out Ivor Benjamin knocked out HSF in mice and found out that they're perfectly fine. They live a normal lifetime. They are more susceptible to certain toxicities, but we were able to take advantage of the fact that they live a normal lifetime to look at this axis between neurodegeneration and cancer. Would HSF play a role? And I'm going to first show you what we've done with cancer. We first asked what would be the effect of HSF on enabling chemical carcinogenesis. So this is a very old cancer model. You shave the backs of the mice, you paint them with a carcinogen and a tumor promoter, and after a while tumors sprout up. And this is what happens in the wild-type mice. What would happen if the mouse just was not able to mount a heat shock response? It's profoundly protective. So this mouse, as I mentioned, lives a normal lifetime. It's, however, susceptible to ischemic injury, or it's susceptible to actually being heat treated in a hot water bath. It's susceptible to toxic stresses, but it is profoundly protected from cancer. So this is the survival curve of these mice. These are the HSF deficient mice. Profound difference in their survival. And this cancer, it turns out, is known to be driven, and in fact we established it in our own hands as well, it's known to be driven by by mutations in the RAS oncogenes. So then we were wondering, okay, what about a completely different way of getting cancer? What about mutations in tumor suppressor genes? So we took advantage of Tyler Jax's P53 model, where he used a mutant P53 that mimics a hotspot in human tumors. And what we found there, too, is that the mice that are missing HSF are profoundly protected. Here we even have an effect in the heterozygote, and while the wild-type mice are highly susceptible to this cancer. So profound effects. So what about humans? Mice don't equal humans, and there have been quite a few things that have not turned out to be, that work in mice and that turn out not to be really effective in humans in terms of cancer biology. And what about maintenance? I've been telling you about the initiation of cancer. What about cells that are already carcinogenic? Do those require HSF for the maintenance of the oncogenic state? And I'm not going to show you a lot of this data. We had published this paper in Cell, I think it was about a year ago. So you can take a look at it if you'd like. But we looked at a wide variety of different cancers, human cell lines that were derived from prostate cancers or brain cancers or breast cancers or HeLa cells or cells that were transformed in culture to the oncogenic state by known oncogenes. And there was just an absolutely amazing effect when we inhibited HSF across virtually every single one of these tumors. The cells started to apoptose. And more recently, which we have not published, we've started looking in tissues derived from people who've had cancer. And we've been doing this, both a pathologist in my laboratory, Sandro Sanagato, and Tan Inche are doing this. And they have looked at breast cancers, and they've looked at brain cancers. They've looked at a couple of gastrointestinal cancers, beginning to look at prostate. And basically, this is staining with HSF, heat shock factor. And you can see in the normal tissue, heat shock factor is out in the cytoplasm, and that's where you'd expect it because these cells are not stressed. They don't need their heat shock factor. It's not activated. But when you move into the tumor, you see that it's just an absolute knock your eyes out, knock your socks off effect. And tumor after tumor is working like this. So we think that this might be both, and the more deranged they seem to be, we've only looked at a few dozen so far, but the more deranged they are, the more intense their need for a heat shock factor. We think it may be both a very interesting marker for defining people who are more at risk for recurrence? Maybe. We think it might also be a very interesting target for cancer therapeutics. So, what about HSF and neurodegeneration? Well, neurodegeneration, in many cases, is also associated with protein folding stress. So, neurodegenerative diseases like Alzheimer's, frontotemporal dementia, Parkinson's, Creutzfeldt-Jakob, ALS, Huntington, they're characterized by pathologists as having really quite massive amounts of misfolded aggregated proteins in them. So, since, as I mentioned to you, this protein homeostatic stress response is conserved from yeast to mammals, and we can go back and forth and look at the kinds of client proteins, all sorts of other things from yeast to mammals, we decided it might be worth trying to take some of these proteins into a yeast cell, see if we could learn anything about the misfolding and the pathology that was caused by these proteins. So I'm going to tell you a little bit about some work we've been doing with alpha-synuclein, which is implicated in a number of different synucleinopathies, several different diseases. You see these aggregates of alpha-synuclein either in neurons or glia. As an example, and probably the most studied of these, is Parkinson's disease, which is a relatively common neurodegenerative disorder. And it's characterized by loss of dopamine-producing neurons from a particular region of the brain, which causes severe motor defects and many, many other problems, many other associated pathologies. And in fact, we now know that there's a much broader pathology in many, many different cell types, but this is the pathology that's perhaps the most immediately obvious and is associated with some of the initial onset symptoms. And there are bewildering variety of different things suggested as being causative for Parkinson's disease. Most Parkinson's disease is sporadic, which means basically we just don't know what the heck has happened. It might be a lot of small mutations of small effect combined with environmental effects of relatively small effect in combination having a big effect. But there are really a bewildering variety of environmental influences, and I just want to mention a couple. One involves mitochondria stress. So things that target the mitochondria can cause early onset Parkinson's. And another one is heavy metal ions. There's been quite a few suggestions from several different types of studies, for example, that manganese exposure causes predisposition to Parkinson's disease. So very different kinds of things. And the hallmark of these different diseases that are called Parkinson's diseases upon autopsy is that you see these blobs of misfolded, mistrafficked alpha-synuclein, this blobs of this protein in the brain. And as I mentioned, it's mostly sporadic, but there are some genetic forms, and importantly, these genetic forms, some of them, actually have mutations in alpha-synuclein. They either have point mutations that cause misfolding of alpha-synuclein, or they could simply have a few extra copies of alpha-synuclein. It predisposes individuals to get early onset Parkinsonism with some slight differences in pathology, but a synucleinopathy for sure. So we know that the misfolding of alpha-synuclein itself and misfunctioning of alpha-synuclein can lead to PD. So we decided to try making a yeast model of this synucleinopathy, not specifically of Parkinson's disease, of course, but of synucleinopathy. And we hooked it up to GFP. I'll just tell you that the GFP has nothing to do with the story. We've done it with and without GFP, but it's a nice marker so you could see where the protein's going. And when we have yeast cells that have one copy of this protein, they're fine. Interestingly, the protein goes to the plasma membrane. And that's quite nice because in neurons, the protein is concentrated at the ends of synapses. And yeast are very similar to neurons, believe it or not, in terms of their protein folding machinery, I've already told you, but also in terms of their protein trafficking machinery. in terms of vesicle trafficking, proteins moving around in the cell that's in very organized compartments, moving from one compartment to the next compartment. One thing that's quite different, however, is that yeast cells do not dock vesicles at the end of at the plasma membrane and wait for a signal to fuse. That process is constitutive. So if alpha-synuclein is doing the same thing in a yeast cell as it's doing in a neuron, we would expect it to actually wind up in vesicles and wind up fusing with the plasma membrane. So that was kind of nice. And I should also mention, I forgot to mention, important, one of the reasons why we decided to go after alpha-synuclein first is that alpha-synuclein is a very small protein. It's 14 kilodaltons. And it binds lipids, and it's prone especially to misfolding when it's not bound to lipids. So we figured those are pretty simple properties. Those are properties that we might be able to replicate in a yeast cell. It doesn't have a whole bunch of different domains that have different partner proteins for interacting, which might not be conserved. It's a very simple protein that's involved somehow in lipid biology. So what happens then if we take this thought that, well, protein folding stress is happening all the time and we can normally cope with it quite well? It gets much worse with stress. Also might get much worse with aging. And maybe the reason why so many of these protein misfolding diseases are diseases associated with aging is because in a neuron that has to live a long, long time and has to traffic proteins and move them from one place to a very, very different places, that they might be particularly subject to problems with protein folding stress. So maybe we could mimic that in a yeast cell by just making a little bit more of this protein. And sure enough, when we just doubled the amount of protein in the cells, massive change in its behavior. So now it's not at the plasma membrane, and instead we have these large foci in the cell. And that's associated with a very big, I guess I don't have that to slide with me. It's associated with a very big change in the biology of the cells. These cells grow perfectly well. You don't notice any different. Well, they maybe grow at just slightly slower rates, but it's really not noticeable. You have to look awfully hard to see it. These cells die when you turn on the alpha-synuclein. And there were many things, when we looked at the cells, they accumulate lipid droplets, which is something that's been associated with alpha-synuclein in mammalian cells and in neurons, and they massively produce reactive oxygen species, which has been associated with many neurodegenerative diseases, including the synucleinopathies. And so we decided, okay, the only reason for doing this in yeast is to be able to do high-throughput analysis, to actually test every single gene in a genome-wide fashion for genes that affect this, to be able to do high-throughput drug screens, and we've done both. I'll just tell you about the genetics. Many of you may know about the fact that in yeast, one thing that's available, and it's very helpful, is a library of deletion strains, strains that have individual deletions in different genes. There's about 4,000 of these strains that you can use, and we've used those. And we've gotten some interesting things out of them. But we've had even better luck with an overexpression screen. So we have a library, and the reason is because the mutant strains are harboring these mutations, and they evolve, and they've changed, and they acquire other mutations, et cetera. But these overexpression strains carry a copy. Each strain will carry a single copy of a different gene. So we've got well over 5,000 of these plasmids that each encodes one individual protein from yeast. And we've got them under the regulation of the same promoter that we were using to express the alpha-synuclein. So these cells are not evolving and changing. We can transform them with both alpha-synuclein and with the gene of interest that we want to query and do all the husbandry of the cells, and nothing is happening. And then only at the time that we want to test the effect of that particular gene on alpha-synuclein-mediated toxicity do we turn them on. And in this particular series of experiments, what we did was we used a strain that had, we dialed down the level of alpha-synuclein a little bit, such that the strain was sick, but it wasn't dying, so that we could look for in the same screen suppressors or enhancers of toxicity. Got a lot of stuff. We got things involved in galactose induction. That was comforting because we're turning these things on with galactose. We should get things that are involved in galactose induction. But of course, that's completely uninteresting with regard to pathology. But we got things involved in protein trafficking, manganese transport. It's turned out that we have had 60 metal ion transporters in our screen, and we got 3 metal ion transporters out of it that's scored in the screen, and all 3 of them are manganese transporters. One transports manganese and calcium, one transports manganese and iron, and one transports manganese and we don't know what else. Nitrosative stress and oxidative stress responses. We've got kinases and phosphatases that are controlling a variety of pathways. We still don't understand them. We've got a few things involved in protein quality control, specifically in the ubiquitin pathway, and some genes of unknown function. Now, the protein trafficking genes were quite specific. We got a bunch of genes that were involved in ER to Golgi trafficking, such that genes that blocked ER to Golgi trafficking greatly enhanced the toxicity of alpha-synuclein, and things that moved it forward suppressed it. Now, all of this turns out to be, of course, of absolutely no use unless we can show that it matters in a neuron, too. So we collaborated with 3 different laboratories to do this, and I just want to say how wonderful these collaborations have been. We've worked with a nematode model, a Drosophila model, and rat embryonic midbrain cultures, primary cultures, with Chris Roche. And I'm just going to show you one result with the rat embryonic midbrain cultures. And I'll just tell you that before we got here, we'd shown the same thing in these other models. So RAB1 is a GTPase, very highly conserved protein that regulates trafficking from ER to Golgi. And we found that overexpression of RAB1 not only rescues yeast cells from alpha-synuclein, it rescues cells that are expressing, neurons that are expressing alpha-synuclein. So in these cultures, Chris typically sees, the way they do their cultures, they typically see about 7% dopaminergic neurons. And when they express A53T, a mutant alpha-synuclein that's associated with human disease, that percentage goes down, so many of the cells in the culture actually get sick. Many of the neurons wind up dying, but the dopaminergic neurons are even particularly sensitive. And we see when we co-express RAB1, it very strongly suppresses the toxicity. And this just shows that we also see Golgi fragmentation, and the Golgi fragmentation is returned to normal. And we have now done this with several other genes from our screen of very diverse functions. And basically what we've found is that we have a lot of suppressors that will suppress the toxicity of alpha-synuclein, proteins with very diverse functions. And this one, this one particular one was not confirmed. We're not sure if it doesn't work or it was just an oddity of the yeast cells, which we certainly expect we're going to get a lot of those, or whether it was just something wrong with that particular construct. But anyway, genes with very diverse functions are showing up as being involved in protecting against alpha-synuclein toxicity in a yeast cell. And those also wind up protecting against alpha-synuclein toxicity in a neuron. And I should have pointed out on the last slide that we've done a lot of controls here. If you overexpress beta-galactosidase, for example, it doesn't suppress toxicity. It's not just if you put in any old protein, it'll suppress toxicity. Many of these we've also tested in nematode models as well, and I just want to point out especially this one here. Oops, I misspelled that. It should be YPK9. It's a yeast gene that we've now renamed from its original name to yeast Park9, and that's because there's a mutation that has recently been identified in a gene called Park9, which is a gene of unknown function in yeast, and it's a gene of unknown function in humans. It seems to be involved in metal transport. In fact, we have evidence that's involved in manganese transport. When that gene is mutated in human beings, it's one of these rare genetic forms that can cause early onset Parkinson's disease. And we have found that when we overexpress that protein function, we suppress the toxicity of alpha-synuclein in the yeast cells. So yeast cells have then therefore allowed us to leap to the realization that this protein, which is actually present in a different compartment, and there would be no reason for suspecting it had anything to do with alpha-synuclein itself at all, is actually directly involved in mediating its toxicity in Parkinson's disease through alpha-synuclein. So we think that this is the tip of the iceberg. We're screening for compounds that suppress toxicity, and some of those compounds also suppress the toxicity in neurons. Interestingly, they also suppress the toxicity of mitochondrial poisons, making another connection between this vast interplay of many, many different features of this complex disease. And we hope this is the tip of an iceberg in another way, in that we're working actually on several different neurodegenerative diseases, and we're very certain that many of those are not going to have any, we're not going to see anything relevant to human biology in the yeast cell. We're certain about that. But we think that in some cases we will, and in those cases we think it's going to be hopeful, because we can use the yeast cells to move very rapidly from high throughput genetics back into neuronal systems. I just want to tell you one more thing about a different model that we have. It's a poly-Q length-dependent toxicity. There are a lot of proteins which when they have little regions of poly-Q in them, and when those poly-Qs expand, they cause a variety of different types of neurodegenerative diseases. This one is actually, yeast cells are actually expressing exon 1 of the Huntington protein, and you can see that the toxicity varies with the length of the poly-Q, which is true in the human diseases as well. Now, we've done full-scale genetic and chemical screens for suppressors of this toxicity. And I just want to tell you one thing, that virtually everything we've seen from the high-throughput screens is different. So it is not, and I think the genetics in the neurons tells you that anyway, but I want to emphasize, it is not just that we've got some misaggregating, misfolding protein and we're getting some generalized protein misfolding stress. We are really mimicking some of the very specific features of the toxicity of these proteins that are related to human disease. So virtually everything is different except, returning to HSF, the heat shock response. Things that turn on the heat shock response in these models, which are otherwise very different, are protective against these pathologies, these toxicities. And, well, what about the heat shock response in neurodegeneration in an organism a little closer to us? How about mammals? There have been suggestions that heat shock proteins might play a role in a variety of different neurodegenerative diseases. And I just want to show you one result with HSF, the heat shock factor that's regulating the whole response. Remember earlier I showed you that those animals were profoundly protected against cancer? This is a mouse these are mice that are dying of prion infections. You've heard of prion diseases, mad cow disease. We've been modeling, it's a protein folding disease, of course, and we've been modeling it in our laboratory too. This is what I would call a train wreck neurodegenerative disease. There's just nothing that can stop this. It's just, when you inject a mouse with prions in its brain, that mouse is going to die. There's just no stopping it. And they die, with very, very different mice. They all die the same time. But when we have a deficiency of HSF, they die even faster. So HSF is having the biology of normal heat shock factor, that normal protein homeostress, is having an effect that's beneficial in this neurodegenerative disease. So this might look as though we are between a rock and a hard place. That this heat shock response, this protein homeostasis, which is balancing us between neurodegeneration and cancer, there might not be anything we can do. I've said that the heat shock response may help protect us from a wide variety of different pathologies to do with protein misfolding that are associated, for example, with neurodegenerative diseases. But on the other hand, the heat shock response seems to fuel the oncogenic profile. So is there going to be anything that we can do? It fuels the oncogenic state. So if we inhibit the heat shock response for cancer, will that necessarily give us neurodegenerative diseases? I don't think so. I really believe that there's great hope. This response is so intricately and intimately tied up with human biology. I've talked about infectious disease. I've talked about cancer and neurodegeneration. I think that there are going to be a wide variety of different ways we can use knowledge of the heat shock response to interfere in those pathologies. And just to give you some examples of our thoughts on this, we think that, for example, for cancer therapy, it might just be a short-term treatment with something that antagonizes HSF is all that would be needed. Not as a standalone treatment, but maybe as an adjuvant to other treatments. And perhaps we could also work with compounds that don't cross the blood-brain barrier to provide some additional protection against the potential of harming the neurons. But it doesn't seem like you really need the heat shock response. Normal people need much of a heat shock response most of the time. As I showed you, mice can get away without it. So another therapeutic strategy might be to find things that potentiate the heat shock response for neurodegenerative disease, rather than outright inducers, which might make the cells in the body more susceptible to oncogenic transformation. Rather, find compounds that might allow us not induce a heat shock response in normal cells, but induce one in cells that are experiencing protein homeostatic stresses, like the misfolding of proteins like alpha-synuclein or Huntington. And in fact, we have found some compounds like that, and they look positive. We might be able to find compounds that don't regulate the whole response, but regulate particular arms of the response, or break into the feedback loop, etc. So I'm really actually very optimistic. We're doing a lot of work in my laboratory right now is aimed at trying to understand how this response, this general proteostatic stress response, figures in a wide variety of situations that matter in human biology and medicine. And we're screening for compounds, we're using RNAi, we're making transgenic mice, and I can just tell you that we have some very promising, at least in these very early preclinical days, compounds. And so I want to maybe stop for just a moment to say, when finishing up here, that I have been profoundly fortunate to have some really remarkably wonderful people working in my lab, people who are not only really smart and really imaginative and really creative, but also wonderful human beings. And I just want to credit the people who helped with these experiments. Kathy and Debbie and Didier and Keith I already mentioned. Young and Mike did the work on the early work on the yeast maturation of Sarc and steroid receptors. This is Leah Cohen. She did a tremendous amount of work on the role of HSP90 in the evolution of antifungal drug resistance. Whoops, I got my thanks misplaced. Tiago and Anil and Aaron did the screen, set up the biology for alpha-synuclein. I want to especially credit Tiago because when we first decided that we might want to try this alpha-synuclein thing in yeast cells, not only did everybody in the outside world tell us we were insane, but people within my own lab were telling Tiago he was insane, that he was just absolutely crazy to try to do this. And he really persevered and did a wonderful job. Julie did the drug screen, which I haven't had time to tell you about. They have been participating in some gene expression analysis, which I didn't have time to tell you about. These folks did the work on Huntington's. And these are wonderful, wonderful collaborators from many different labs around the country. I've mentioned them most by name when I showed their experiments. They've been just absolutely fabulous. Science is really changing, I think, in terms of it becoming a really collaborative science. Those people helped with the screens, and this is Luke Whitesell and Cheng-Kai Dai, who did the work on HSF and cancer in mice. And Sandro, I mentioned, he's been working on the pathology and showing the staining of HSF in the nucleus has jumped into the nucleus and oncogenically transformed cells from a human tumors, and our collaborators on compounds. And I think that's, and Ivor, who gave us the mice that were deficient in HSF, and Tan Inche, who's another pathologist, has been, and it was actually his slide that I showed you from the human breast. I think that is actually all. Okay, thank you very much.
So one would assume that the heat shock response is important for survival of aneuploids. Is that Is that or is that not correct?
Heat shock response is important for survival of
Aneuploid, cells that are aneuploid, that have departed in chromosome.
Angelica Aman has done some very nice
Right.
work on that, and aneuploid cells are expressed in the heat shock response.
Right.
I'm not sure that she has yet shown that it's required for their survival.
Okay. So I guess the follow-up to that would be, to what extent is the sort of tumor protective properties of the heat shock, of HSP, of the heat shock response acting just because cells tend to go massively aneuploid before they form tumors?
We don't think it's working just because of that, and the reason why we don't is that we've drilled down in this paper that we published. We have some of the work, but we've drilled down into several different aspects of it. What we think is happening is it's actually affecting lots of aspects of cancer biology in a relatively small way. It's actually discouraging apoptosis. It's promoting a variety of signal transduction pathways, like, for example, using the HSP90 protein. It's changing the balance, helps to shift the balance from respiration to fermentation. It helps cells to, let's see, what are the other aspects? To become more susceptible to RAS-mediated transformation and other oncogene-mediated transformation. It is a very, very multifaceted, and there are many, many different proteins involved in it, but each of them seems to affect maybe a two-fold or four-fold or five-fold effect on any one of these different processes, and it's together that they have their big effect. That's a good question.
I have two questions on the role of HSF in cancer. So the first is, about 10 years ago, there was a paper from the lab of Rick Morimoto where they showed that the activity of HSF actually decreases with aging with the increase of HSF binding protein, which inhibits HSF activity. So whereas the tumors would tend to increase with aging. So how would you fit that?
I'm sorry, the sound is just not very good. I'm not hearing you very well.
So
it's kind of muddled for some reason.
Rick Morimoto's
Yes.
lab showed that HSF activity decreases with aging about 10
Yes.
years ago, whereas tumors tend to increase with age. So how would you fit that with your model that HSF is needed for tumorigenesis?
Because as you saw in that tumor, when we looked at what is happening with HSF, its basal activity may be declining a little bit. We don't know. It's out in the cytoplasm. I don't know if you'd noticed that. But in the tumor, there are things that are actively turning on the activity of HSF. So we actually find that in a lot of different tumors, in addition to the translocation of HSF from the nucleus, from the cytoplasm to the nucleus, we also see that there's somehow the cells are especially for the more deranged cell types, they're actually expressing higher levels of HSF as well. And we see HSF bound to chromatin in the transformed state. And it correlates, at least in the relatively small number that we've looked at, with how profoundly oncogenic they are. So for example, with HeLa cells, there's a tremendous amount of HSF, and it's all in the nucleus. And if you inhibit HSF in those cells with RNAi, they die right away. So, I don't know. HSF function may well decline in general as we age, and that may be, that would make sense that we then, therefore, are more susceptible to neurodegenerative disease. But there are a lot of other things going on that initiate cancer. I'm not saying that, in fact, I should explicitly state, HSF is not an oncogene. So if you overexpress HSF, it doesn't itself drive cancer oncogenesis. What it does is it participates as in what's so-called non-oncogene addiction, by which cells become addicted to HSF because it helps fuel the biology of the transformed state.
And the second question is about the conservation of this role. So heat shock response, as you showed, is conserved even to plants and to yeast. But plants, let's say Arabidopsis has about, 21 different HSFs, doesn't get cancer or anything at any stage. So what do you think about the role of HSFs in plants? In this
Cancer,
pathway?
I don't know. Depending on the plant, they have different numbers of HSFs. Even mammals actually have 3 different HSFs. The HSF that I told you about is the one that seems to drive most of the stress-induced HSP70 The others are developmentally regulated, heat shock proteins. The others are more developmentally regulated. So we know there are, for example, in terms of the plants, I don't really know, but I'm going to guess that a lot of those HSFs might be developmentally regulated. We showed a long time ago that, for example, this is also very highly conserved, in seeds, in yeast spores, in, what else did we look at? We looked at gametes of Drosophila. So after meiosis, with meiosis, for some reason, cells turn on high levels of these heat shock proteins. And that's driven by HSF, too. There's a specific HSF that's involved in the expression of heat shock proteins in sperm. So I think that there's a lot of, I'm just giving you the tip. I really believe that I'm giving you the tip of the iceberg and sort of trying to paint a picture that this response is involved in a lot of our biology. It's very ancient. It's not really, when you think about it, it's not surprising that it's involved in so much of our biology. It's this very general stress protective. But that also is involved in developmental biology as well, because the spores, if you deplete heat shock proteins in a yeast spore, it sporulates just fine. But they become very sensitive to stress. So yeast spores will normally survive anything. But if they don't have even one individual heat shock protein like HSP104, they'll die. We also showed that Arabidopsis, actually, as an example, HSP101 is made stored in seeds, and those seeds are quite resistant to heat, and even the early germinating, very first germinating plants have some of this protein, and they're protected. As soon as that protein starts to decline from those plants, they become very sensitive. So there's a lot of other biology out there, too.
Thank you.
Neurodegeneration as well as cancer is, to a certain extent, is related to the age. So the question is there a differential turnover of heat shock proteins in neurons versus epithelial cells?
Yeah, an interesting question. In general, it seems as though neurons don't mount as strong of a heat shock response as you'd expect them to mount with a heat shock. And in fact, we have found with cells that are accumulating exon 1 glutamine expansion Huntington protein, they've got massive amounts of misfolded protein, and yet they're not mounting much of a heat shock response. And that's why I think trying to find potentiators that would cause those cells that are already experiencing protein folding stress, but are for some reason not doing what they're supposed to do and turn on a heat shock response, maybe we could find things that would make those cells particularly sensitive. And as I mentioned, we have some compounds that we think do just that.
Does it get worse with age?
Does which get worse with age? The ability to, the loss of a heat shock response? That I don't know. People have worked on that and have suggested that it's true. I just don't know that worked very well.
Thank you very much, Dr.